Great. Okay. So I'm going to talk about phytoplasma diseases with talking specifically about the Bogia coconut syndrome. So just an outline of what we'll talk about: phytoplasmas generally, palm phytoplasmas and how bad they can be, and then what can be done to control them. And that's all before I talk about the Bogia coconut syndrome. And it might sound like you're putting the cart before the horse, but no, it's not the case. You'll understand why I'm doing it that way as we go through it. Research done and what still needed to be done. So what are phytoplasmas? They are wall-less bacteria, and hence that's a very old electron micrograph photo on that slide, showing they're like this amoeboid kind of shape as they move through the phloem vessels of plants and squeeze their way through the sieve tubes and things like that. The vectors are these larger insects that can feed on phloem, mostly leafhoppers, plant hoppers, and psyllids. Most phytoplasma symptoms are caused by the hormonal disruption of the plant. So you get weird things happening to the flowers and the growing points, witches' brooms and little leaf symptoms. So those photographs on the left are witches' brooms and things of some weeds in Australia, a witch's broom of Melaleuca in the middle. And the photographs on the right are from the Pacific Islands. That's a tomato big bud symptom, I think, from New Caledonia. In the corner, bottom right corner, is a strawberry not being a strawberry at all because it's been totally stuffed up by the phytoplasmas, and that's also from New Caledonia. And at the bottom is a cassava witch's broom from Wallis Island in the French territory of Wallis and Futuna. So just to illustrate that they're all over the place. And so what we're focusing on here is the kind of, not those symptoms, but the kind of interaction between phytoplasma and plants that results in rapid, sudden death. And these are the new encounter diseases.
Richard, just before you go on, can you just, there's a little app thing that you can hide in your, can you see that? You can just press hide. I
didn't see that. I'm looking at the other screen, sorry.
Oh, that's perfect now. Go for it.
Didn't notice that. Okay. So phytoplasma diversity, because they can't be cultured, it's a really insanely complex naming system has happened. They're classified into groups and subgroups based upon their 16S ribosomal RNA gene sequences. And so far there are 39 groups, so they're labeled with these Roman numbers, 1 to 39. And the important thing here is that in our part of the world, Oceania, Pacific Islands, Australia, Southeast Asia, etc., mostly they're group 2. Group 2 is all over the place. And so far they're given these Candidata species names because they can't be cultured. And so far there's more than 50 names have been proposed. And there's a list that you can get on the internet there. And they changed the guidelines recently so that to become a member of two phytoplasmas, become the member of the same species, they have to have more than 98.65% identity of that length of DNA that is acknowledged as the standard. I'm not going up and down anymore. Can you see the next slide now?
Yes, we can. Perfect.
Yes. Sorry, my up and down button stopped working, but I've got it now. Differentiation of the different phytoplasmas started off with breaking up this PCR amplified fragment of DNA using enzymes to get like a fingerprint for each different groups of phytoplasmas. In the old days, this was done with gels, with polyacrylamide gels. And like with so many things in this world where you used to mix toxic chemicals on the laboratory bench, they found a way to do it with computers now. So there's a US Department of Agriculture website, which can do all that digestion in the computer and produce these sort of patterns you see on the right. Then, of course, there's the sequence analysis of PCR amplimers, which is so important. And more now it's moving to whole genome analysis. It's getting more and more complex and more differentiation is happening. The palm phytoplasma diseases. I just want to emphasize the history of this. So the lethal yellowing diseases of palms were absolutely devastating last century and probably before that even in other parts of the world. So in the Caribbean, Africa, Florida, Mexico, to both sides of the African continent, there was absolute massive devastation of coconut palms. So these, and I say, it became very famous news at the time. So that's something from the New York Times in 1977 and the National Geographic in 1988. This really was big news. And I'd like to emphasize here that much closer, all of those phytoplasmas were assigned to groups 4 and 22. We also have other phytoplasma diseases present in Malaysia, Indonesia, Sri Lanka, even one from a Pacific island, which is unpublished data, which are in other groups. And the important thing is they're much, much less damaging diseases, and they shouldn't be lumped together. And there's some literature out there which does lump them together. Honestly, it's like lumping gastroenteritis and the Ebola virus as saying they're almost the same as each other. It's not the case. We need to separate them, and the lethal yellowing diseases are the most important ones. So the new additions to the really bad family are the latest numbers in the numbering system, group 38 and 39. So one is the Bogia coconut syndrome in PNG, and the other one is something we found in Cairns in the last few years in Australia. And I'd like to emphasize that those photos are from Cairns, and the coconut palm on the right, that melted down in a few weeks, just like they did in Jamaica, just like they do in Africa, just like they did in Florida. And that's important because I'll show in the next slide. So they are in that really bad family. This is a phylogenetic tree from a publication just a few months ago. And so you can see that the 22, the 4, the 38, and the 39, they all sit there related to each other in the same part of the tree. They really are in the same bad family together. Here in Australia, I suspect we just don't have those massive plantations to allow the epidemic to explode. And that could be a vector insect thing. PNG, though, is a different story, and we'll get onto that in a minute. Management options. There's a lot of interesting work done in the Caribbean last century, towards the end of last century. And basically the management was relying on things like host resistance. They identified alternative hosts where the other plants that the phytoplasma is infecting. And there's a nice example that people talk about from the 2000s in Jamaica, the Michael Black plantation, which is just a study test case that really reduced the losses, as you can see on the screen there. And it was based upon very strict surveillance and removing the infected plants, and they burnt them, and that's an important thing. Weed control, so once they knew what the vector was, they could take out the grass that its nymphs feed on, which is very important, and other things like planting poor vector breeding hosts and ground covers. And that photograph in the corner is to remind me to mention that you shouldn't create one problem by dealing with another. And that's a coconut rhinoceros beetle affected palm tree from Port Moresby. You can't just cut those palms and leave them lying around, which I'm sure was probably discussed at the last webinar. So, Bogia coconut syndrome. It's just as devastating as those historical diseases. Up there in that part of Papua New Guinea, there's hundreds of thousands, maybe millions of coconut palms have been destroyed. So it was a quiet problem, bubbling away quietly towards the end of the last decade or so. Politicians took notice in the early 2000s, and the first positive phytoplasma detection was in 2008, and that was a joint work between our organization and PNG's NACIA, the two quarantine organizations. And then there was an ACR project that ran from 2013 to 2019. And so I'd like to emphasize here, I wasn't on the ACR project, but I did have a lot of involvement at the beginning and the end, and other interactions along the way. Just to mention sampling, the best thing to do is to get a chainsaw and cut down the coconut tree and dissect out its heart. That's the most high phytoplasma titer, but obviously that's really difficult, and you can only do a few trees in a day. So another alternative way that's almost as good is to take drill tissue, which the photograph of us on the right, drilling lower down the stem, and there's enough phytoplasma in that internal tissue to get a detection. So what are the insects spreading the disease? the ACR project made some really, really great advances with this work. And they were using a method whereby you get the insects to actually feed on a sucrose solution. And that way you prove that the insect actually it's not just a chance contamination that the phytoplasma is present in its gut or on its mouth parts or whatever. It's actually multiplying in the salivary glands and coming out of the saliva and into this sucrose solution, which is tested by PCR. And so that photograph, the yellow photograph, is just a little mock-up we did for a workshop. That's actually a plastic insect in there. That's the method they used in PNG. And then the other photograph is from a more recent publication showing another way of doing it. And the insects implicated, and this is all work that has been published in the, everything I'm talking about here is actually public information available on the ACR website at the final report of the project. And they identified the two insects at the top are probably the most important, Sophiuma and Lophops, also flattids, dervids, and ricanids. They did cage experiments. So basically they put these cages up in a real hotspot of coconut infection, and those insects that have been identified from the sucrose experiments, they basically put a photograph of each one, that photograph on the right, a photograph of each one on the entrance to the cage, and over a period of many months, they just collected the insects that looked like the photograph from the wild, were placed inside the cages to see if they could cause disease, phytoplasma disease, in the target plants that were inside. So the results were not fully conclusive. A lot of factors beyond people's control. The insects were very hot. The insects died rather than multiplying. They fed and died. And many of the coconut palms in there died. Towards the end, as you can see in the photo on the left, the plants started poking the roof. And so insects from the outside actually fed on the controls and caused some infection. So it wasn't a perfect experiment, but it really went a long way towards indicating what's going on. And I think they managed to, they very strongly implicated, or they showed transmission of the Lophops and the Sophiuma. Alternative hosts is the other thing you need if you want to tackle this disease. And you can't talk about the coconut disease if you don't talk about the banana disease at the same time, banana wilt-associated phytoplasma. This is the symptoms of banana wilt disease caused by phytoplasmas in PNG. That really strong yellowing, discontinuous vascular streaking inside the pseudostem, and if you can find the male flower, the male bud rather, the male flowers usually rot. And this is a map of, there's one thing I want to emphasize here is that this is a map of the different BWAP phytoplasmas that have been identified, and this will be published very, very soon in a book chapter. So the different dots are the different variants. So the banana wilt phytoplasmas are actually quite a variable family. So the green dots on the right, which are in Bougainville and the Shortlands Islands in the Solomon Islands, that they are so different from the Candidatus phytoplasma novaguinense that they will be in a new species of their own one time. The others belong to Novaguinense, but they are variable within that. And so the darker dots, we called that one the Debbie Parry phytoplasma, after the village in PNG where we first found it many years ago, 2015, I think, 2011, I think it was. And that one is the most common one in PNG. And I just want to put it out there, we can talk about that later. We're getting some circumstantial evidence that even though this one fits within Novaguinense, the Candidata species, it seems that it can't kill coconut palms. Just circumstantial evidence, no proper experiments. It's something that needs further work. Sorry, the other thing I meant to emphasize there, the palms and coconuts are only dying up there in Madang province. Everywhere else in PNG and the Solomon Islands, there is no palm disease whatsoever. It's just the banana phytoplasma disease that's showing. Sorry, that's a very important point I should have emphasized. Other alternative hosts, as we know, in these countries, there are many vegetatively propagated crops and things that are important. So the ACR project team sampled 180 odd plants, and they got a very high, plant samples taken from disease hotspots, and they detected the Novoguinense phytoplasma in a high number of taros and a few other things: kunai grass, sweet potato, cacao, African tulip. And to my mind, taro is the absolute key finding here in terms of biosecurity, because what do people do when they travel somewhere and they bring back some taro that tastes really good? You cut off the top and you plant it. So you instantly transported all the pathogens that are vegetatively propagated from one end of PNG to the other. So that, to my mind, is a really, really serious issue that needs following up. So that was all before COVID, before 2020, the ACR project finished up. And then we had an opportunity to go back just a few months ago in November last year. And this was a very big joint operation, I guess, between Australian institutions, University of Queensland and Queensland Department of Agriculture. And then also NARI from PNG and KIK and NBPOL, New Britain Palm Oil, who was originally Ramu Agri Industries before. And NARI's given me permission to talk about this unpublished data. It's just still a work in progress that we're still doing, but we followed up and collected 230 similar samples from hotspots, other different plants, and they've all been tested by real-time PCR, and so far we've got no positives at all. They're all negative. And that's 60-odd taros. That's the other kind of taro, not the Colocasia esculenta, the Xanthosoma species, the Dalo nitana in Fiji, Dalo fortuna, I think, if he's in Tonga. Different names for different taro. So we still haven't been able to follow up on the true taro and investigate its host status. But we found quite a few kunai grass and a bunch of other things in that list below. Seed transmission was another very important aspect of the ACR project study. So originally thought not to happen in phytoplasmas, but there's more reports turning up recently. And again, I guess the bottom line here is the jury is still out. There's evidence of possible seed transmission from the experiments conducted by that project, but there still needs some more follow-up. So I'll just leave it there. That's what's written in the current wrap-up report for the project. Evidence of possible seed transmission in coconut and betel nut. So this is the NARCIA set up a roadblock to try and limit the spread of the disease right at the beginning of this whole episode. And hopefully they might have covered many of the issues there, because that sign is saying don't carry bananas, taros, or yams for planting out of that area. So even though there's an issue with taro, maybe it might have been dealt with to some extent already. So the final slide really is just to mention or reiterate what still needs to be done. So what the growers of PNG, coconut growers, need is integrated pest disease management strategy. And for that, they'll need more work on the biology and ecology of the insect vectors, find out what the vectors really are, what do they feed on, et cetera, et cetera. More phytoplasma host range work. What do the insects feed on? Epidemiological studies on how the coconuts react with the phytoplasma, and resistance or tolerance. And there's a germplasm collection. It was 1 kilometer away from the disease front when we visited last year, but eventually this germplasm collection at the Stewart Research Station in Madang is going to meet the phytoplasma disease head on, and it'll be a ready-made disease resistance trial. So there'll be some very useful information there about what's resistant or tolerant. And then more work on the genetic diversity of the group of phytoplasmas. These are a group of monocotyledonous infecting phytoplasmas, which is highly complex in bananas and palms. And the photograph I've put at the bottom there is just a little reminder that when we did our travel last year, one of the members of the team from Madang, Morobe province, found these Ophiuma and said, "Oh, I've never seen this where we work. This is a new insect to us." So maybe that's the missing link. Maybe the insect distribution might be the real answer as to what's happening. Thank you very much.
Thank you so much, Richard. That was fantastic and really nice to actually go through the sort of story from the introduction right through to an example case study there, and really fascinating. I mean, I have lots of questions. There's also a question you've already answered, but that was from Myanmar around, is the phytoplasma disease, could it be carried by seed? So I think you've sort of addressed that, and that it could be, but there needs to be a lot of further work to find out more. I'm also going to, I'm going to put Carmel on the spot soon as well, because I know that she's been working on this. She's shaking her head, but I might ask her, and I'm going to give you a bit of time though to think of something, but I would be quite keen to just have some feedback around what SPC or what's happening in the region, if there's any examples of some of that follow-up work, I guess, that Richard has sort of pointed out as possible things to focus on in the future. Before I ask you that question, Carmel, I'm just going to ask Richard a couple of questions. You talked about sort of, I guess, more focus on that whole genome analysis as well, or I guess understanding a little bit more what's happening there. Is that work being done by anyone, or do you know of that research, who's leading that research or where it's being done? Because that could be quite interesting, obviously, as well, at sorting out some of those unknown factors.
Yeah, the University of Queensland people who were participating in that project, or that little trip that we made, one of them is a PhD student specifically doing that work on the banana phytoplasma. There was certainly a lot of discussion during the ACR project about doing similar work on the coconut phytoplasma. I'm not sure how far that went. There were a number of samples were collected and returned to Australia with a memorandum of understanding with NARI, and they were coconuts and bananas. So there's a lot of scope there, and certainly that laboratory, that group at the University of Queensland are certainly in a position to do a lot of make a lot of advances with that work.
Excellent, thanks, Richard. Another question here is, what's the risk of these higher impact coconut diseases like this one you're talking about in Papua New Guinea spreading outside Papua New Guinea? I mean, you sort of talked about the need for strong quarantine and thinking about how it's spreading, but is it high, low, medium? I mean, how risky is it for the region?
Well, at the moment, it's been totally confined to that one area, and that was because of government intervention. Nakia was involved, and they set up a roadblock and public awareness and all that kind of thing. And every time there's a report of coconuts dying outside of that region, then the Nakia guys are in there straight away and taking samples and testing. So it's been really well under control so far, but it does worry me that if it's transportable in popular staple crops like taro, that is really, you know, that's really raising alarm bells. You know, like I mentioned, people. I've seen it myself with my own eyes. People from Port Moresby who will go out to the country will always return with fresh veggies because they're much cheaper and better quality than what they can find at home. And as I say, you know, if you taste a good taro, you're going to plant it. That's what people do.
Yeah. And so there's obviously a lot of research focused in that PNG, maybe Solomon Islands area. So are other countries in the region, are they actively screening for these phytoplasma diseases? Do you know of other?
Well, I think there's and Carmel will be able to comment on this, there's certainly a high awareness within SBC, which has been conducting plant health surveys in the region in recent years. And my own experience, I worked for SBC about 20 years ago. We did a lot of virus and virus-like disease surveys. So we collected a lot of phytoplasma samples, and definitely back then, coconut phytoplasma diseases were a #1 target. We were watching out for them all the time. So I think we can rest assured that there's not a bubbling outbreak somewhere, because these things don't hide away. We're not in a place where there's coconut plantations. The one in Cairns is the exception. It's really not spreading very much. But why
all of a sudden, though, the one in Cairns? I mean, what's the imbalance or the thing that suddenly this comes as a problem? Same in P&G, I guess.
Yeah, I really don't know. It was an accidental discovery. The botanical gardens contacted us and said our palms are dying, and we looked at them and we said, oh yeah, it's probably a fusarium. Here's the address of somebody in Sydney you can send a sample to. And by the way, we've been working on this thing in P&G. We'll just test it for phytoplasmas just to eliminate that possibility. And we were absolutely astounded when we got these positives. And then we investigated more and found it was widespread in a small area of Cairns, about 5 to 8 kilometers square. Many different ornamental palm species, and palms here die of other things like Ganoderma, especially in the dry season, and perhaps people just weren't noticing. And I suspect we don't have a really efficient insect, and we obviously don't have coconut plantations to allow massive epidemics to build up.
Okay, that's not reassuring though. What you've just told me, I have to say, it's quite frightening. I'm going to throw it over to Carmel very soon. She's going to follow up on and maybe give us a bit more detail. But here's a question here and feel free if you don't, you can answer it. But on average, how large is the whole genome sequence? Closer to viruses or bacteria? It's an easy or hard thing to compare isolates. Or is it an easy thing to compare isolates through whole genome sequencing? You can.
I have to admit that stuff goes over the top of my head.
No, that's okay, because you've got lots of other stuff in your head. But let's leave that question, because I'm sure we can find someone, and even Carmel might be able to help us. We'll see, or we'll find out somebody.
A more informed opinion of that sort of thing.
Okay, just another couple of quick questions, then I'm going to throw it over to Carmel. But here's one here. Does rainfall help with the spreading of phytoplasma?
Rainfall. Rain. Well, you would think that it helps the insects to grow and multiply and spread. So when everything is growing well, as opposed to a dry season, etc. So I would have thought so, yeah.
Okay, well, I'm going to throw that, Richard, I'm going to thank you very much. I'm going to throw it over to Carmel to add on any other observations that you have. I know you're an expert, so don't be too shy here, Carmel. So tell us what's happening a little bit more if you'd like to add. From the Pacific's perspective or from your own perspective of within the broader region. You're just on mute, Carmel.
Yeah, sorry, your general question on phytoplasmas in the Pacific. I think the last work that was done was actually when Richard was there doing his surveys. And so we haven't really, I don't think, I've only been with SPC 4 and a bit years, but I think between the time that Richard was there and later on, there weren't many surveys done in the region for phytoplasmas. Basically, it's been okay, if something is wrong, somebody goes to investigate and check. So as far as we know, we haven't received any reports. I'm not working in the plant pathology area. I'm managing projects at the moment on coconut. But as far as I know, we haven't received any new reports of phytoplasmas or phytoplasma type symptoms from countries in the region on coconut. Yeah, so in terms of the coconut projects that I'm working on, of course, our aim is to bring some of coconut embryos from the different countries into CPACT and then be able to do tissue culture, multiply those and share those within the region because there are our regional members looking for new varieties of coconut. We have a large diversity of coconuts in the region. And of course, there's a danger there that we may be exchanging infected material. Of course, we're building capacity in CPACT, in the germplasm health unit, to be able to test for phytoplasmas and BCS in particular, because we are intending to get some materials from PNG. And so while Richard talked about his trip last year, we actually were sampling at the same time that Richard was in PNG, and we just could not meet up. So, you know, our trips sort of overlapped and we just didn't manage to meet up with him. But yes, we are looking at working in with the CIRAD team, and all of the samples that we tested were positive for coconut. We tested heliconia, and we also tested banana, and the banana and coconut were positive. The heliconia was negative, and surprisingly, although we saw symptoms in betel nut, they were all negative. So this is, so some nice results there. And we're hoping that we could build on that moving forward because of the association, as Richard knows, between banana and coconut. We think that it might be easier to control the disease in banana rather than coconut. So perhaps if there is an interaction there, if we can control it in banana, breed clean, in tissue culture, develop clean materials by treatments in tissue culture and distribute clean banana material, that might help to reduce the incidence of the phytoplasma in banana populations as well as coconut, initially in Madang, of course. So we're looking at, we're hoping that we can get some funding to develop that work with different partners as well.
Fantastic.
So that's sort of where we're at with the BCS. But yes, we will be, the issue of transmission in embryos, of course, is not proven. Well, not totally. We've had a few results from Jeff Gur's project, the ACR project that Richard mentioned, and also from some work in Mexico in tissue culture plantlets, where they have found, in Carlos Oropez's lab, they have found some phytoplasma in some of the tissue culture plantlets. But there are ways, like as with viruses, to eliminate these in culture, in tissue culture. And I think that's moving forward. That's perhaps a new development we can look at into trying to clean these materials up, vegetatively propagated materials especially, before redistribution, as we do at CPACT already for viruses. So viruses are cleaned by various treatments, and then they're distributed to our members. So
well, fantastic. Thanks for the explanation. You're very good at thinking on the spot, because I really did put you on the spot, everyone. So well done. Thank you very much. Really interesting. And I have to say, there's also a lot of work going on, there's some work going on in Southeast Asia, and particularly South Asia as well. So we've got some experts, I think, in this room, and also that are very interested in doing similar sort of work, also from Sri Lanka and India, that I've been in contact with. So thank you very much to Richard. Thank you very much to Carmel for adding on to that. Great presentation, really interesting, and I think it's something that we need to keep a close eye on, because obviously we don't know sometimes when these things pop up, and getting a really good handle on that, and lots of strategies to cope with it are very important. So thank you very much. And I'm going to move on to our next speaker, Dr. John Rand.